The overall goal of this protocol is to describe how to use laser microirradiation to induce different types of DNA damage, including simple strand breaks and complex damage, to study DNA damage signaling and repair factor assembly in vivo.
In the 1960s and 70s, we focused a laser beam through a microscope that you can see behind me and was able to damage a small region of a chromosome. The cell divided, survived, and we couldn't understand why. Well, 40 years later, we are now using this method to selectively study DNA repair, and that's what you're going to see today.
This method can help answer key questions in the DNA repair field, such as how DNA damage is recognized and processed, and how damage signal propagates in the cell.
Laser microirradiation allows for real-time, high-resolution, single-cell analysis of macromolecular dynamics in response to DNA damage confined to a submicrometer region within the cell nucleus.
However, various laser conditions have been used without appreciation for differences in the types of damage induced, causing inconsistencies in cellular responses, factor recruitment, and modifications.
To express fluorescently tagged proteins in HeLa cells, on day 1, seed 6 to 8 times 10 to the 4th cells in 0.5 milliliters of culture medium in one well of a 24-well plate. Then place the plate in the tissue culture incubator. On day 2, perform liposome-mediated DNA transfection by diluting 0.3 micrograms of a mammalian expression plasmid DNA encoding a fluorescently tagged DNA repair factor.